Hello everyone. So now in this video we are going to discuss about the fluid mosaic model. I need not say what is this. We all have studied about the plasma membrane or the cell membrane, right? So what is fluid model? What is the fluid mosaic model? How it is related to the plasma membrane? This is the most recognized, most modern theory or model, you can say, which explains the structure of the plasma membrane most accurately. Now, who has proposed it? You already know. The name is, see here, S.J. Singer and G.L. Nicolson in the year 1972. Now, our body contains nearly 37 trillion cells. But the real mystery is, how does each cell decide what enters and what leaves the cell, right? Oxygen enters the cell for respiration, nutrients enter the cell for respiration, but the waste that leaves the cell. So oxygen enters, nutrients entered, waste are leaving, but the harmful substances are blocked. So who actually controls this microscopic security system? Who is in charge? The answer is the plasma membrane, right? This is the outermost membrane in case of the plant cell. No, this is the outermost membrane in case of the animal cell. But in case of the plant cell, outside the plasma membrane, they have additional layer that is called cell wall that we are going to discuss in the subsequent videos. So plasma membrane is the outermost layer in case of only in the animal cell. Don't do the mistake. Now, today we are going to discuss about the fluid mosaic model, which is the most modern explanation given by Singer and Nicolson, which I already told you. Now, see, there were some previous models also which tried to explain the structure of the cell membrane, such as the lamellar model, the Robertson model, the Danieli and Dawson model. I repeat, the lamellar model was there, the Danieli-Dawson model was there, the Robertson model was there. Then why we need another one? Why there was a need of another one? Because they could not explain the structure properly. The explained structure, the theory that they have proposed, and the functions were not matching. So actually, you know what happened? The earlier scientists, they thought the membrane was just a simple static layer. That was the biggest misconception they had. They thought it's a simple static layer. But what happens? But the experiment shows that something strange. What happened? The proteins could move sideways inside the membrane. Yes, the proteins could move. And the membrane was flexible, dynamic, and constantly changing. So scientists realized the membrane is not rigid, it behaves like a fluid. Right? We already know what is the word, English meaning of the word fluid. What is the meaning? Fluid means something which can flow, right? So here what happens? The proteins are moving sideways. So obviously we can say it's a fluid. Why mosaic? Because the proteins and lipid are alternately arranged. So that's why the name, that is fluid mosaic model. Now, so If you look in this diagram carefully, you can understand the plasma membrane or the cell membrane. Now, if you zoom little bit, see, it's a fluid phospholipid bilayer. Obviously, the proteins are floating like the icebergs in a sea of lipids, right? Now, we are going to discuss about the structure of the phospholipid. Which are the phospholipids? Here you can see. So each one of them, see here, look carefully, each one of them are the phospholipids, right? So actually it is made up of the phospholipids, then there are proteins. So these are the proteins, right? These are the proteins. Now what is the structure of the phospholipid? Look here. The phospholipid has a polar head, which is called the hydrophilic head, and they have a hydrophobic tail, which is made up of fatty acids. It's an amphipathic molecule and arranged in a bilayer. You can see the bilayer, right? Polar and non-polar, with hydrophilic heads facing outwards and the tails facing inward. So that is the basic structure of the phospholipids. Hydrophilic head towards the outside and hydrophobic tail towards inside. Then what else we have? We have this. We have the cholesterol here. Then the proteins. Now here is the important part. The protein could be two types, integral and peripheral. Which are the integral proteins? See, these are the integral proteins. And peripheral, these are the peripheral proteins. Right? Why? Because the integral proteins are actually, they are the transmembrane proteins, but peripheral proteins are not. Now, next is the carbohydrate part. You can see the carbohydrates, they are attached to protein. They are called as glycoproteins. Here you can see the glycoproteins. Here, the glycoproteins. Also, there are glycolipids. Here you can see glycolipids. So glycoproteins are nothing but protein and carbohydrates. Glycolipids are lipids and carbohydrates. Alright. Now, as we now understand that the phospholipids has two phases, right? One is the hydrophilic, another is the hydrophobic. Now the hydrophilic, the word philic means, affectionate or attracted, something like that. So they are on the outer side, both the sides. You can see the blue color here, if you look carefully, here also, here also. But at the center, the hydrophobic tails are concentrated. Why? Hydrophobic, phobia. It comes from the word phobia. When they are scared, they don't want to face the water, they're scared of water, right? So that's why they are clustered at the center. If you look in the electron microscope, here if you look under the electron microscope, you could see the dark bands, the protein layers, lipid layers, and again the dark bands, which are the protein layers. So all the light bands or the lipid layers are at the center. They don't want to face the water. They're hydrophobic, phobia, right? Okay, so this much we understand. Now the question comes, why it is called fluid? Because I already told you, it is called fluid because the phospholipids and many proteins can move laterally within the membrane. So this is clear. Next, types of protein we have already discussed here. See, types of proteins, two types, integral and peripheral. Now the most important part, what is their function? They help in transport, carrier functions, works as a receptor. The peripheral proteins remains loosely attached on the membrane surface. Can you identify peripheral proteins? I just told you, right? Peripheral proteins are loosely attached on the surface. They mainly help in enzymatic activity and cell signaling. So the peripheral proteins. Which are the peripheral proteins? Look carefully. Which are the peripheral proteins? So these are the peripheral proteins, right? These are the peripheral proteins. What they do? Enzyme activity, cell signaling. Then we have the cholesterol also. You can see the cholesterols. What do? They are present between phospholipids. They work like a membrane stabilizer. Remember this. The cholesterol acts like a membrane stabilizer. It actually prevents the membrane from becoming too rigid in cold conditions or too fluid in high temperature. I am repeating, the cholesterol acts as a membrane stabilizer. It stabilizes it. Right? Why? Because otherwise the membrane will become too rigid in cold and too fluid in the high temperature. It should not be like that. It should maintain a consistency, and cholesterol helps in that. We already studied about the carbohydrates, two types, glycoproteins and glycolipids. What is their function? Their function is cell recognition, attachment, antigen formation and communication. Now, selective permeability. This word is very much associated with the fluid mosaic model. Selectively permeable or selective permeability. Why? Because the membrane allows only selected substances to pass through it. Yes, it's very choosy. Achha. Next topic is membrane asymmetry. What is this? Membrane asymmetry means, see, the two sides of the membrane are not identical. The carbohydrates are mostly found on the outer side. See, this is the outer side. Outer side. This is inner side. Right? You can see on the outer side the carbohydrates are mostly concentrated, but inner side it is not. That is actually mentioned as the membrane asymmetry. I hope you are understanding the importance of this, right? It's a fluid mosaic model. It is fluid in nature, mosaic because of the presence of the proteins and lipids. The main functions, it protects the cell. It creates a boundary, right? It creates a boundary. Then you have the, see, if this is the nucleus and other organelles are also present, but this is the boundary. This is the boundary that is maintained by the cell membrane, and we are explaining that with the help of this fluid mosaic model. It helps not only the boundary but transport, communication. It also maintains the internal environment. So we can say the membrane is not just a boundary, it's an intelligent dynamic control center. This is very important. It's a intelligent dynamic control center. So always remember, the plasma membrane is a dynamic. Again and again the word is coming, dynamic. It's a dynamic phospholipid bilayer with floating proteins, cholesterols and carbohydrates together forming the fluid mosaic model. Thank you.